Cells use various methods to transport molecules into and out of them so as to maintain cellular functions. Passive transport is membrane transportation that does not use energy. An example of passive transport is diffusion. Diffusion is the movement of molecules of a substance so that there is an equal concentration of the substance throughout the available space. The unequal concentrations of the substance form a concentrating gradient, and the substance will diffuse down its concentration gradient from areas of high concentration to areas of low concentration. Facilitated diffusion uses protein channels and carrier proteins to aid diffusion across the membrane. This allows larger molecules and charged molecules to cross the cellular membrane. Facilitated diffusion does not use any energy. Water moves into and out of the cell through the process of osmosis. Osmosis is facilitated by specialized channel proteins called aquaporins. Like in normal diffusion, water moves from areas of high concentration to areas of low concentration. Low concentration areas are called hypotonic, while high concentration areas are called hypertonic. A cell suspended in a hypotonic solution can explode due to a rapid intake of water. A cell suspended in a hypertonic solution can rapidly lose water, shrivel up, and die. A cell is at isotonic equilibrium with the surrounding solution when the concentration of water in the cell is equal to the concentration of water in the solution. At this point, water will move at equal rates into and out of the cell. Active transport allows substances to move against their concentration gradient with the use of carrier proteins. This allows cells to maintain concentrations of certain substances even if the cellular environment differs from the external one. Energy is required to move the substances against their concentration gradients. Exocytosis is the process of expelling materials from the cell with vesicles at the surface of the cell. The secretory vesicles fuse with the membrane in order to discharge the materials that it is carrying. In plant cells, exocytosis is important to forming the cell wall, while in animal cells, exocytosis is important for the excretion of hormones, neurotransmitters, and endocytosis is the process of the cell engulfing biological molecules with a vesicle, which is formed from the cell membrane as it pinches in until it is completely detached from the membrane. The sac will then transport and release the enclosed material. Phagocytosis is the process of a cell engulfing particles and transporting them into the cell with the use of a special vesicle called a vacuole. The vacuole will eventually fuse with the lysosome, where the particle will be digested. Penocytosis is the process of a cell engulfing extracellular fluids with vesicles. This allows the cell to gain large quantities of solutes that are located in the extracellular fluid. Receptor-mediated endocytosis allows the cell to intake large quantities of extracellular molecules at once, regardless of their concentration outside of the cell. These substances, called ligands, bind to receptors on the cell membrane. Once enough ligands have bound to the receptors, the membrane will form a pit and engulf the ligands and other molecules. The new vesicle will then take the substances into the cell.